As you know, the separation of two strands of DNA duplex is required for the replication to start. But DNA polymerases, they are unable to separate the two strands of duplex, although they are able to add new nucleotides to the single strands, but they cannot separate the two strands. Therefore, another class of enzymes, which is called DNA helicases, they catalyze the separation of the two strands of duplex DNA at the replication fork. So at the replication fork, DNA helicases are present which open up the strands. DNA helicases are hexameric proteins. That is, they contain 6 types of subunits that assume the shape of a ring. So this DNA helicase, it is in the form of a ring, and 6 subunits are present in this ring. This ring encircles one of the two single strands of DNA at the replication fork and adjacent to the single-stranded double-stranded junction. So here it is a single-stranded double-stranded junction, and just adjacent to this junction, this helicase, it encircles one of these two strands. Here in this diagram you can see this is the replication fork, and adjacent to this single-stranded double-stranded junction, this is also called a single-stranded double-stranded junction. So adjacent to this junction, this DNA helicase is present, and see, this DNA helicase is only present on one of the two strands. It is not present on both the strands. And if you see the three-dimensional structure of this DNA helicase, you can clearly see there are 6 domains or 6 subunits in this protein. So this is a hexameric protein, and it is present in the form of a ring. DNA helicases found at the replication forks exhibit high processivity because they encircle the DNA. They associate with the DNA and unwind multiple base pairs of the DNA, which means they have a high processivity. Release of the helicases from the DNA therefore requires the opening of the hexameric protein ring, which is a rare event. This ring cannot be opened. If it is opened, it can release the strand, but it happens very rarely. However, the helicases can dissociate when it reaches the end of the DNA strand. It encircles a DNA strand, and it keep on moving as the replication fork keeps on moving. When this replication fork reaches at the end of the DNA duplex, so both the strands will be opened up and this ring can exit this strand. So only there is one possibility, that once the replication is completed, this ring can leave the DNA strand. Of course, this arrangement of the enzyme and DNA poses problems for the binding of the DNA helicase to the DNA strand in first place. So because this is a ring form, so how this ring encircles this DNA strand first time? Once it encircled this strand, then it will keep on moving, and at the end of replication it will leave the strand. This is this can be explained, but how for the first time it encircles this strand. There are some specialized mechanisms that open the DNA helicase, this hexameric ring, and place it around the DNA before reforming the ring. So there are some special mechanisms which do that. Each DNA helicase moves along single-stranded DNA in a defined direction. This direction is also defined. Either this will move in this direction or this ring will move in this direction, that is either from 5 to 3 prime or 3 to 5 prime. This property of this helicase is referred as the polarity of the DNA helicase. DNA helicases can have a polarity either of 5 prime to 3 prime or 3 prime to 5 prime. This direction is always defined according to the strand of DNA bound rather than the strand that is displaced. One of the strand is displaced and the other strand is that which is bound to this DNA helicase. So the bound strand will define this direction. So as here you can see in this diagram, this is the replication fork and this hole is a replication bubble. In this replication bubble, this is 5 prime and this is 3 prime. Similarly, this is 5 prime and this is 3 prime. So this strand will decide what will be the polarity of DNA helicase. Either it will come here and the polarity will be 5 to 3, and if it will bind this ring or this helicase will bind here, the direction will be 5 to 3 prime. So this is the polarity of DNA helicase.